So hello everybody. Professor Leo Eckhart, my co-chair, and myself are really thrilled to have you with us today at this new session of the ESDR Kitchen. For those of you who are not too familiar with our kitchen, so this is actually a series of live events that the ESDR has been organizing for the past few months to try continuing promoting scientific communication during those challenging times. So we have four different sub-series of Even in the Kitchen, the recipe book where new techniques are being presented, the sweet and sour series where two protagonists are discussing a usually controversial topic, or molecular cuisine where leaders in their field share their personal journey through scientific discovery, and finally, the freshly baked series where recent and groundbreaking discoveries are discussed, and this is our session today. So I'd like now to pass on to Leo, who will introduce our two speakers of today. Leo.
Thank you, Eli. So it's my great pleasure to introduce today's speakers who will present two exciting papers that were recently published in the JID and in Nature Aging. As always, please send your questions and comments during the talks using the chat or the Q&A function. And the questions to both speakers will be discussed after the two presentations. So the first speaker, the first presentation comes from the Côte d'Azur. Unfortunately, we are only with the speaker virtually. Speaker is Dr. Meri Tulic, who is a researcher with INSERM, working in the team co-directed by Professor Thierry Passeron and Dr. Stéphane Rochy in Nice. And the primary focus of her research is vitiligo, in particular the role of innate immunity in the initiation of the disease. And today, Meri will present her work published in the JID, and we look forward to your presentation. Please.
So, good afternoon. And before I begin, I would like to thank the entire ESDR Kitchen team for giving me this opportunity to present to you our latest baked publication, looking at the analysis of matched skin and gut microbiome of vitiligo patients and the link with mitochondrial and immune changes. Oops. Sorry. As we know, vitiligo is an autoimmune disease characterized by skin depigmentation due to the loss of melanin-producing cells called melanocytes. Although JIVA studies have shown variation in more than 50 genes to be associated with the disease, the delay in the age of onset over the last 30 years emphasizes the role of environmental factors in triggering vitiligo. We've recently shown that the stress and activation of innate immune cells triggers the initial melanocyte destruction in vitiligo. We also know that bacteria are among the top producers of stress signals, or PAMPs. There is now a large body of literature showing gut dysbiosis to be associated with skin disease, and more recently, dysbiosis in the skin has been associated with skin disease such as atopic dermatitis, acne, and psoriasis. Here we set out to examine the role of skin microbiota in vitiligo. And to date, there is only one paper in the literature by Ganju and colleagues who have looked at only surface microbiota in vitiligo skin without comparing it to the healthy skin. A more recent study by Caroline Leopold's group in a mouse model vitiligo have shown that antibiotic-induced depletion of bacteroides in the gut may induce skin depigmentation, suggesting a possible link between gut and the skin. So the aim of our study was to phenotype gut and skin microbiota in vitiligo patients and to look at the difference between lesional and non-lesional sites, as well as the differences between surface and deep microbiota in the skin. And finally, to compare this microbiota in vitiligo skin to microbiota of the healthy patients. This was a fairly intensive study for our patients where we obtained more than 6 samples from each subject. We had 10 controls and 10 vitiligo patients. We took not only the surface skin swabs, but also deep skin biopsies, as well as gut stool samples so sampling different sites. We also took skin samples from both non-lesional and lesional sites, sampling different zones, and we collected blood samples to be able to examine changes in microbiota with immune function and disease severity. So initially looking at the gut microbiota, this slide represents different measures of alpha diversity within the sample, looking at the number of species, their diversity, and their richness. Results have shown reduced alpha diversity in vitiligo samples compared to healthy controls, demonstrating for the first time changes in the gut of vitiligo patients with skin disease. Here we see composition and diversity of our samples by comparing relative abundance of bacteria at phylum level in 10 control and 10 vitiligo patients. We see a trend for increased Firmicutes in purple and decreased Bacteroides in green, resulting in a significant increase in FB ratio between the two groups. In the literature, this increased FB ratio has strongly correlates with obesity and cardiovascular disease, as well as other autoimmune disease. Interestingly, it's been shown that Bacteroides is protective in the gut of mouse vitiligo model, hence its loss may contribute to the onset or exacerbation of the disease. So to examine if there are differences between swab and biopsy samples from the same patient, we measured alpha and beta diversity in each of the paired samples and have shown that both the number and the diversity of bacteria were higher in the swabs compared to biopsies. And we saw significant differences in the beta diversity between biopsies and swabs, which allows us to conclude that microbial diversity and richness differs in relation to the sampling method. The most obvious difference is that while in the swabs, more than 30% of the genus belongs to the Staphylococcus, while in the biopsies, it is the Bifidobacterium, which makes up more than almost 60% of the total genus, suggesting that certain genera correlate strongly with the collection method and that there is a need for simultaneous sampling of surface and deeper skin to get an accurate picture of changes in the skin microbiota, which has important implications for interpretation of our results. So how did these results differ? Initially looking into the swab data, although our results have shown increased alpha diversity between control and vitiligo samples, we see no significant changes between non-lesional and lesional regions in the vitiligo. Examining the composition of these samples, first we see significantly decreased Firmicutes in orange in lesional compared to non-lesional sites. And this is most likely due to the significant reduction in Staphylococcus shown here in blue at the same site. We've also seen a trend for increased Proteobacteria in green in lesional compared to non-lesional site, which is a bacteria containing many known human pathogens related with inflammation. So moving on to the skin biopsy samples. In contrast to the swab, we now see differences in alpha diversity, not only between control and vitiligo patients, but also between non-lesional and lesional vitiligo samples. In lesional samples, we see decreased diversity, but increased community richness. And importantly, in contrast to the swab microbiota, we've demonstrated that the lesional biopsies shown here in red had the most distinct microbiota compared to all other groups. The increased richness can be seen in these taxa plots showing lesional biopsies to have decreased actinobacteria in orange and increased Firmicutes in purple. However, the largest and the most significant difference is the presence of Tenericutes, which is shown here in pink, and mitochondrial DNA in brown, exclusively in 7 out of the 10 lesional sites. This was very interesting because there is reports of interaction in the literature between mitochondria and microbiota, and which has implications in ROS production in vitiligo cells. And in mice, we know that high mitochondrial DNA has been linked to reduced microbiome diversity. So looking at the genus taxoplot, the most prominent observation is the almost complete absence of the blue bifidobacterium in the 7 out of the 10 lesional sites. In lesional biopsies, what we see is a general depletion of the commensals such as bifido, Shigella, Staphylococcus, while there is an enrichment in the pathogenic bacteria such as Mycoplasma and Streptococcus. And this was seen in the same 7 patients who had shown strong increase in mitochondrial DNA. To try and understand why these 3 patients behave differently than the rest, we looked at the immune function, because we know that the presence of mitochondrial DNA has been linked to the loss of bifidobacterium, which is an important protective commensal and has been shown to decrease the activation of innate immune cells. Therefore, we hypothesize that it's the increased mitochondrial DNA and or loss of the bifidobacterium in the lesional skin, which activates innate immune system. To test this, we measured serum levels of innate and stress markers and have shown that 7 patients with increased mitochondrial DNA in the lesional skin, so the patients 4 to 10, had significantly increased immune function compared to the patients without elevated mitochondrial DNA, so patients 1 to 3, suggesting that mitochondrial damage at the lesional site is associated with peripheral increased immunity and stress in the same patients. So in summary, we've shown that there exists both gut and skin dysbiosis in vitiligo patients, and that although sampling skin surface is easy, it gives us limited information, and sampling deeper layers is more informative to study skin microbiota, at least for the vitiligo patients. Using biopsies, we've been able to show that lesional skin has a unique microbiota compared to the other sites, and that the dysbiosis in lesional skin is associated with the depletion of protective bifidobacterium, enrichment of mitochondrial DNA, oxidative stress, and immune activation. So to conclude, here we've shown a comprehensive phenotyping of skin and gut microbiota in vitiligo compared to healthy subjects, and have shown a link between skin dysbiosis, mitochondria, and immunity in vitiligo skin. Now, whether these changes are a cause or a consequence remains to be confirmed, but nevertheless, it gives us some new therapeutic targets to examine for potential treatment of vitiligo. And finally, I want to thank my whole team, particularly Thierry for his relentless support, and my talented PhD student, Anen, who did most of this work. And of course, ISIS Pharma for funding the study. And I'd like to thank you for your attention.
Thank you, Meri, for this very interesting and exciting presentation. The paper will be discussed after the second presentation today. So the second speaker is Dr. Emma Chambers. Emma graduated in immunology from the University of Bristol and subsequently undertook master and PhD studies at King's College in London. And during her postdoctoral research in the lab of Professor Arne Akbar at University College London, Emma investigated how cutaneous immunity changes with age. And in 2020, Emma was awarded a Bart's Charity Lectureship to join the Center for Immunology at the Blizzard Institute, where she has established her own group investigating, again, aging and immunity. And today we are happy that Emma will present her recent paper in Nature, Aging. And Emma, thank you very much.
Thank you, and thank you for inviting me to talk today. So our lab focuses on looking at cutaneous immunity, antigen-specific immunity, and more specifically looking at how it changes with age. And the way in which we do this is we inject antigen intradermally into individuals' forearms, and then we look at the immune response afterwards, and we can really study what's going on within the skin, by either doing punch biopsies over the antigen injected site, or by forming suction blisters over the site of challenge and then taking the cells out from the blister fluid. So the antigen that we're focusing on for this talk is varicella zoster virus antigen, or VZV, which is a causative agent of chickenpox and shingles. And we know that the incidence of shingles increases greatly as we age, so we think it's an important target to study. And so what we do is we inject, just going to change it to pointer. We injected antigen intradermally, then 2 to 3 days later, we look for the immune response in the skin by looking at redness, the size of the response and the palpability, and give it a clinical score. And here we have a typical response from a young person, and here we have a typical response from an older person. And when I say older, I mean anyone over the age of 65 years. And as you can see, we've done this on a number of people. And generally, when you have a younger person, they have a nice big response to the antigen, and as such, a high clinical score. And conversely, when you inject antigen into an older person, they have a much lower clinical score, at times 0. And the clinical score really correlates with the immune infiltrate. So the higher the clinical score, the more immune cells are present within the skin, showing that it's a good antigen-specific immune response occurring. As ever with humans, there's always exceptions to the rules. So we have some young people who have very low response, and conversely, an amazing older person who have a fantastic immune response. As part of our studies, we wanted to look at, do some RNA-seq analysis on VZV injected skin, and we used saline as our control. And what we got was some really unexpected results. In the older people, as shown on the right here, they had a large pro-inflammatory response to saline injection 6 hours after the injection, which was not observed in the young. And we really wanted to follow up this data to understand more. What we observed is that in those individuals who had a very high inflammatory response to saline, they had a very poor clinical score. And conversely, in those individuals that had a good immunity to VZV with a high clinical score, they had no inflammatory response to saline. So it somehow suggests that this inflammatory response to saline is negatively correlating with a VZV specific immunity. And to cut a long story short, some bioinformatic analysis identified that the cells that seemed to be responsible for it were mononuclear phagocytes. There are some remaining unknowns from this study that we wanted to follow up. Firstly, which population of mononuclear phagocytes were recruited to the skin? What was the mechanism for their recruitment? And then importantly, how do these inflammatory cells block antigen-specific immunity in the old? And then actually the real important question is, can this be reversed therapeutically? Can we make an older immune response look younger again? So to answer the first question, we looked at macrophages, dendritic cells, and neutrophils, and we saw no change in the overall cell number before or after saline injection. However, when we look for monocytes, and to do that, we use the markers CD14 and CCR2, we observe in the older people that there's a significant increase in CD14 positive and CCR2 positive, CD14 positive inflammatory monocytes at the site of saline injection, which was not observed in the young. To confirm this data, we did multiparametric flow cytometry to confirm that it was these monocytes that did increase. And again, we saw this significant increase in these inflammatory monocytes in saline injected old skin, but not in the young, as shown here in white. So the first question we had was this a salt to saline specific response, or is this actually a response to needle challenge in itself? And to answer that, we injected the same volume of air into the arm of older people and younger people to see if we also see this increase in inflammatory monocytes. And indeed we do. When we inject air into older people, we see this significant increase in this inflammatory monocytes, which was not observed in the younger individuals. So the follow-up question is, do we see the same response in response to antigen challenge? And indeed, yes, again, we do see this significant increase in inflammatory monocytes at the site of antigen challenge. So it suggests that older individuals are responding to wound damage in an inappropriate way, generating this inflammatory response. So the next question is, which cells were responsible for recruiting these inflammatory monocytes to the site of antigen challenge and saline? And we went back to our microarray data and looked at a number of chemokines that are responsible for recruiting monocytes. And we found that the majority of them were increased in saline injected skin as compared to normal in older individuals, but we didn't see it in the young. We decided to focus on CCL2 because it was one of the most differentially expressed genes and one that's key in recruiting inflammatory monocytes to the site of challenge. And we confirmed our data by immunofluorescence, where we saw this significant increase in CCL2 positive cells in saline injected older skin, but we saw no difference at all in younger skin. So to cut a long story short, we know that older individuals have an increased number of senescent fibroblasts in their skin as compared to younger individuals. And we also know that CCL2 is a component of the SASP, the senescence associated secretory phenotype, which is this inflammatory phenotype that senescent cells possess. So we wanted to identify if it indeed was the senescent cells were excreting CCL2 and recruiting these inflammatory monocytes into the skin. And we identified senescent fibroblasts using P16 and FSP1. And we did indeed find that there was a significant increase in senescent fibroblasts expressing CCL2. So the next question is how are these inflammatory monocytes inhibiting antigen-specific immunity? So we went back to our microarray data and we looked at a number of pathways that could be involved in inhibiting antigen-specific immunity. And you can see again in saline injected older skin as compared to normal, they were generally increased. We decided to focus on this one, PTGS2, because it's the gene that encodes COX2. And it was one of the most differentially expressed genes in saline injected skin. And again, we confirmed by immunofluorescence staining that there was indeed a significant increase in COX2 expression in saline injected skin as compared to normal skin. And also importantly, there was a significant increase in these monocytes expressing COX2 as well. So to confirm that these monocytes which express COX2 have the ability to inhibit T cell proliferation, we went to an in vitro culture system where we co-cultured monocytes expressing COX2, which were generated with LPS treatment for 3 hours with T cells, and compared them to just monocytes, untreated monocytes with the T cells. And as you can see here in the representative data and in the cumulative data, when you have monocytes that express COX2 in the presence of T cells, there was a blockade of proliferation and a significant reduction in the number of cells that go through cell division, suggesting that these monocytes do certainly inhibit T cell proliferation. To confirm that it was COX2 that was responsible for this blockade of proliferation, we added in a specific inhibitor, NS398, which blocks COX2 expression. And with the addition of this NS398, we now get a restoration of proliferation back to the baseline level. And the cumulative data can be seen here. We know that COX2 is upstream of a number of lipid mediators, but we decided to focus on PGE2 as it's one that is known to be associated with blocking antigen-specific immunity. And to assess if PGE2 was responsible for blocking the T cell proliferation, we used a MF498, which is an inhibitor for the PGE2 receptor on T cells. And again, when we add this blocker of PGE2 signaling into the system, we see a restoration of proliferation. So this data suggests that inflammatory monocytes come into the skin and block T cell proliferation by production of PGE2. This experiment was repeated in skin-derived T-resident memory cells, and they indeed were also suppressed by these COX2 expressing monocytes. So the final question, can we make an older person's immune response look younger again? So as part of ongoing study within the group was using this anti-inflammatory drug called losmapimod, which is a P38 MAP kinase inhibitor, which blocks inflammation in older people. So just to give you an overview of the study, we recruited people who were over the age of 65 with no pre-existing inflammatory diseases. We injected antigen, we waited 48 hours, we looked at their clinical score. Presuming that they looked like a typical old person, in that their score was 3 or lower, they continued into the study where they had a washout period, and then they were given losmapimod treatment for 4 days, and then we repeated the clinical score. At the same time point, we were also able to collect skin biopsies of antigen challenge or saline challenge skin, and also blood pre and post the losmapimod drug. And so here I'm showing you the saline data. So we can see again, when we look normal versus saline injected skin of these older people before losmapimod treatment, we see this significant increase in these inflammatory genes. And then importantly, after losmapimod treatment, the inflammatory gene signature is greatly reduced. When we looked by immunofluorescence in saline injected skin, we see a significant reduction in CCL2 production, a significant reduction in fibroblasts expressing CCL2, and a significant reduction in the inflammatory monocytes recruited into the skin. So this implies that if we block inflammation by using this losmapimod drug, we get a significant reduction in the inflammatory monocytes recruited. So then importantly, if we've blocked these inflammatory monocytes, can we now restore cutaneous immunity in these older individuals? The short answer is yes, but I will show you why. So we had 42 individuals who took part in our study, and we looked at their VZV score, so their antigen-specific immune response to VZV, pre and post losmapimod. And as you can see here, there was a significant increase in VZV clinical score in the older people after losmapimod treatment. And when we looked at antigen injected sites 7 days after injection, so you can see pre-losmapimod, there were very few T cells at the site of antigen challenge. So this is CD4 T cells, and the bottom one is CD8 T cells. And then after losmapimod, there's really quite a marked increase in the infiltrate at the site of antigen challenge. It makes them look like they're having a younger person's immune response. So we've got a nice infiltrate of CD4 T cells, CD8 T cells, dendritic cells are shown in blue, and also the cells are proliferating. So this leads us to this model where we have normal old skin. You've got the endothelium, the senescent fibroblasts, the T resident memory cells, and the monocytes. If you inject anything into these older people, air, saline, antigen, it results in this CCL2 production from the senescent fibroblasts, activation of the endothelium, and then 6 hours after the injection, you then got a monocyte recruitment into the older skin. These monocytes express COX-2, they secrete prostaglandin E2, which binds to this receptor on the T cells and blocks proliferation. Really importantly, if we block the inflammatory course using losmapimod, we have a reduced CCL2 production, which means reduced monocytes recruited. I didn't have time to show you that losmapimod also is P38 MAP kinase is upstream of COX-2, therefore administration of losmapimod also blocks COX-2 and reduces PG2 production. And now we have a restoration of proliferation, and we're now able to see that the older people's immune response is able to respond to antigen challenge again. So this work was carried out in the lab of Arnie Akbar, as shown in the middle here, at University College London, with the help of many individuals, as you can imagine, but a special thank you to Melitza and to Hugh, and to all our collaborators at UCL and around. And thank you for listening to my talk.
Thank you very much, Mary and Emma. Really great talks. Just before proceeding, I'd like just to mention that this episode is sponsored by Almira and supported by BMS. So we have a few questions here. I start with Mary. The question here, are there any relation or any form of correlation between gut and skin microbiota changes in what you have been presenting?
Yes, actually, we have shown that was the first lot of results, which was quite surprising, that patients who actually have a skin disease, we can actually see changes in their microbiota in the gut. Now, which may suggest that there is sorry, I'll start my video, which may suggest that there is perhaps a gut-skin axis for vitiligo. However, this really needs to be examined thoroughly, and we've just scratched the surface here with our study. So it's certainly an interesting aspect.
Okay, and then we have a question for Emma here. Why do we see more severe insect bite reactions in the young patients, while herpes zoster is much more severe in the elderly?
Yeah, I think that's due to numerous things. So we're just looking at a secondary immune response or a memory immune response. And we know that, for example, that the T cells are less functional in older individuals, which is why we see this a reactivation of viruses that have been controlled by T cells. Why insect bites are worse in the young and less in the old? It could be due to the fact that actually older skin baseline has a lot of immunoregulatory pathways. But in reality, I don't know the answer to that.
Okay, and then we have another question regarding the skin microbiome. Is the absence of Bifidobacterium, Shigella, and Staph due to the absence of macrophages, inflammation, or others?
I think the question was absence of melanocytes.
Melanocytes, excuse me.
Yeah, no, that's okay. I don't think so, because we've shown that also, certainly in our previous work, we know that these inflammatory markers are upregulated in not only lesional skin, but in non-lesional skin. So, which is obviously where the melanocytes are present. So it may be a contributor, but I certainly don't think that this is going to explain the melanocytes are going to explain the story.
And then a question for Emma. You showed that there were those patients who were like the young patient who had very low levels of response, and the contrary for older patients. So are those exceptions also showing the same kind of profile in terms of gene expression and immunophenotyping as their normal counterpart, regardless of age?
Yeah, so actually in a figure I left out, we look at old responders, as we call them, so old people who have a younger immune response, and they don't have an inflammatory response to saline, and they therefore don't have the recruitment of CD14 positive monocytes. So it looks like that is the case. Yeah, in that, if your immune response, the immune response decreases differently in individuals. So just some young people, some older people just have a fantastic immune response that behaves young. And actually they've been really interesting people to study, to see what is, different with them. And then how can we use that knowledge to then help the majority of the older population.
I think we have time for just one last question. So, Mary, do you consider the difference of skin microbiome population in lesional vitiligo skin taken from different skin areas? So, how do you control for those differences?
No, absolutely. That was the first, when we tried to find an answer why those 3 patients responded differently to the rest. That was obviously the first questions that we wanted to ask. So we thought it was something to do with difference in age, a difference in the site where the biopsy was taken, or perhaps the activity of the disease. However, none of those could explain it. And so it was only when we started digging a little bit further, we found that in the literature, there is this clear relationship between mitochondria and immune responses. And so this is why we decided to look. And much to our surprise, even in such a small number of patients, we saw such clear differences in responses. And I didn't show the data for the interest of time, but we do have a strong correlation between the presence of mitochondrial DNA in the lesional site and the patient's own chemokine and cytokine responses in their serum.
Okay, thank you very much. So I'd like, first of all, to very much thank Mary and Emma for really a wonderful session. I'd like to invite you all to our next ESDR kitchen episode, which is due June the 13th. Birgit Lane and Pierre Colomb will debate about the truly relevant roles of keratins. So thank you very much for having been with us, and goodbye.
Goodbye.